Dr. Sage here, back to talk about the host defenses. In this video, we're going to discuss the development of lymphocyte diversity. By the end of this video, you should be able to summarize the maturation process of both B cells and T cells, draw a diagram illustrating how lymphocytes are capable of responding to nearly any antigen imaginable, outline the process of clonal deletion and clonal selection, and describe the structure of both B cell receptor and T cell receptor. Markers found on T cells. These are antigen-specific T cell receptors. CD3 markers surround the T cell receptor and assist in binding. CD4 binds the MHC class II molecules, whereas CD8 is found on cytotoxic T cells and binds the MHC class I molecules. T cells constantly circulate between the lymphatic and general circulatory system. They migrate to specific T cell areas of the lymph nodes and spleen, and it's estimated that 1 billion T cells pass through the lymphatic and general circulations per day. Hundreds of millions of distinct B cells develop as a result of gene modification and selection. They circulate through the blood, homing to specific sites in the lymph nodes, spleen, and other lymphoid tissues where they take up residence. Have immunoglobulins as surface receptors. So if we're comparing B cells and T cells, B cells mature in the bone marrow, whereas T cells mature in the thymus. Both B cells and T cells have distinct CD molecules, but B cells have immunoglobulin and T cells have T cell receptor. The general functions of B cells are production of antibodies to inactivate, neutralize, and target antigens, whereas T cells function in helping other immune cells, suppressing, killing abnormal cells, hypersensitivity, and synthesize cytokines. Each naive lymphocyte bears an antigen receptor that recognizes a unique antigen. Stem cells become granulocytes, monocytes, or lymphocytes in the bone marrow. Lymphocytes become either B cells or T cells. B cells stay in the bone marrow, whereas T cells migrate to the thymus. Each builds a unique antigen receptor. Both B and T cells migrate to secondary lymphoid tissues. By the time the B and T cells reach the lymphoid tissues, each one is equipped to respond to a single unique antigen. The specific B cell receptor is composed of 2 heavy chains, 2 light chains. One light chain is bonded to one heavy chain. The 2 heavy chains are bonded to each other with disulfide bonds, and this creates a symmetrical Y-shaped arrangement. So the receptor has antigen binding sites. These are pockets at the ends of the forks formed by the light and the heavy chains. They can be highly variable in shape and fit a wide range of antigens. They are the variable or V region, which is found in antigen binding sites. Its amino acid position is highly varied from one clone of B lymphocytes to another as a result of genetic reassortment. Then they have constant regions, where the amino acid content does not vary greatly. T cell receptors are similar to the B cell receptor. They're formed by genetic modification, has variable and constant regions, it's inserted into the membrane, has antigen binding site formed by 2 parallel peptide chains. Unlike the immunoglobulins, they're relatively small and never secreted. Immune tolerance involves tolerance to self, or removal of any potentially harmful clones through clonal deletion. Some autoimmune diseases are thought to be caused by the loss of immune tolerance, the survival of forbidden clones, or failure of other systems. This has been your brief overview of the development of lymphocyte diversity. Until next time, this has been Dr. Sage.